Hello, everyone. My name is Lynn, and for Journal Club today, I will be presenting RNA-targeting CRISPR-Cas-Rx system that relieves disease symptoms in Huntington's disease. First, I'll be talking briefly about what Huntington disease is. It is a fatal neurodegenerative disorder. It's a genetic mutation that is caused by an expanded CAGVP in the Huntington gene. So everybody has a small amount of HTT gene. It normally is harmless, but more than 39 CAG repeats usually cause to toxic protein accumulation, and that leads to brain cells degeneration in the striatum parts of our brain. This toxic protein buildup usually has symptoms such as uncontrollable movements, cognitive declines, and mood changes, just to name a few. Huntington disease, or as you'll hear me calling it HD for short throughout the presentation, it is autosomal dominant. So that means just having one mutated copy is enough to cause the disease. This makes it an ideal target for gene-based therapy. However, simply just reducing the mutant HTT gene has been a big challenge because as of now, there's still no cure for Huntington's disease, and any treatment we have is not long term, and it's only meant to like manage symptoms. So I think we briefly talked about CRISPR-Cas9, but scientists discovered that it is a natural immune response used in microbes to defend themselves against viruses. Scientists are now exploring it as a gene editing tools, creating double-stranded breaks, or even making any changes in RNA. It works with a guide RNA, which tell it where to go on a specific consensus sequence. And the Cas9 protein is like a molecular scissor that will cut this protein out. So CRISPR-Cas9 has been used in other studies, and it showed that it does knock out the HTT gene in Huntington disease. But the problem is that if it cuts at the wrong site or if there's any mistake, it permanently changes the genome, which is pretty dangerous and irreversible. So introducing CasRx, it targets the RNA only. It is smaller and is more precise. It's shown to have lower off-target risk as well. So that means that it's removing these mutant genes without causing any changes to our DNA. So it's reversible, it's safer and viable. Um, and currently, it has been the biggest safety improvement in gene therapy. Um, so why do we want to reduce HTT at the RNA level? Um, a lot of studies or clinical studies have shown that silencing HTT mRNA can reduce symptoms because what's happening at the molecular level is that this mutant HTT RNA is causing abnormal protein aggregation, and that interferes with our cell function. So that means that if we're able to reduce these RNA messages in advance, that could be the key to the gene therapy. Um, However, before the study, scientists weren't sure if CRISPR-Cas9 system would work in larger animal because it's only been done in mice. And there were limited studies that tested to see if CasRx would reduce HTT at different stages of Huntington disease. Because like mentioned before, this disease does progress as we age and because of the accumulation of the toxic protein. So this uncertainty is what the researchers wanted to address. So that brings me to the purpose. The purpose of this experiment or study is to test whether CRISPR-Cas9 system would safely and effectively reduce mutant HTT RNA in one larger living animal and two at different stages of HD, and whether or not these reduction would improve symptoms. So specifically, what we're focusing on in this study is we want to see if it will improve motor function. Will it protect neurons from degeneration, or will it normalize any gene expression pattern that were disrupted? So they use three major lab techniques. First one being Western blotting. The purpose of it is to see how much HTT protein is present. I included the diagram of like the process of each of these tests so we can pause and take a look, but I'll kind of go through it. So Western blot quickly is we run the protein through a gel that we add antibodies that sticks to the HTT gene, and we just observe how much protein is present. So in theory, if CasRx works, what we want to see is a lighter band, which means less protein. They also use RT-qPCR, which measures the concentration of the HTT mRNA. So again, if CasRx works, we want to see lower concentration of HTT mRNA. Lastly, they did immunofluorescence, which is just visualized that activates using glowing antibodies. And at the end of each of these diagrams, we can kind of expect what kind of result we'll see later on. So aside from those tests, they're also going to do an animal study. So first, they use stereotaxic injection to deliver CasRx directly to the striatum of these mice at different ages, 3 months, 6 months, and 9 months of age. And similarly, in pigs, we'll be observing untreated pigs versus treated pigs. And on top of that, we'll be doing behavioral analysis. So in mice, they'll be conducting a rotarod test, which is shown in the picture here. It's just a rotating rod, and we just want to see their latency to fall from it. And they also perform pole climbing tests and balance beam tests just to observe their motor function. And in pigs, they just go through treadmill test and a footprint tracking, which is conducted on like sandy path, so we can observe their coordination and balance. So the results in mice, the first figure is what we got from the Western blots. They're comparing the results between the control group, which is unaffected by Huntington disease. The middle is the treated with the CasRx, and lastly, untreated. The first band shows the expression of our mHTT, or mutant HTT gene. So we do see a decrease in the expression in 6 months compared to 3 months, and 9 months compared to 6 months. In the PCR, we do see a decrease in mHTT expression as well in treated mice. And in figure 3 is our immunofluorescence staining, just to quantify the number of aggregates. I think specifically they want us to focus on the red, which is actually the protein aggregates we're looking for. So in untreated versus treated, we do see less aggregates showing. So in neuropathology and behavioral analysis, as mentioned in Huntington disease, it does progress with age. So we do see more motor deficits at 6 months, and then a lot more motor impairments at 9 months. However, HD mice that was treated with CasRx did perform better on those motor tests than untreated mice. Um, Additionally, they also looked for a certain gene expression, just to see what kind of gene is being suppressed and what's activated in treated mice. So in the suppressed gene now we're seeing they're suppressing cell killing, T cell activation, response to virus. So just a lot of immune reaction often seen in HD, and more genes is being activated like phagocytosis, vitamin metabolic, spinal cord development, which is very important. Um, so some of these genes regulation is being kind of partially restored as well. So in pathology, in Huntington disease mice, the striatum normally shows some increased glial cells. This process is called gliosis, and those cells are your astrocytes and your microglia. The increase in these cells usually correlates with injury or disease. And so in treated mice, we see a decreased number of these cells, which is good because that means it is reducing the neuropathology of Huntington's disease. And in the first graph, that is showing the marker of neuron. And we can see that it is consistent with all three different test groups. Um, that is important because it means that it didn't harm any healthy neuron, which is huge because it's avoiding any neurotoxicity. Okay, as for results in pigs, these are kind of backward. So we're looking at immunofluorescence first. It does show a decrease in MHTT aggregates as we can see in the little arrow that they included. We're seeing a lot less dots and aggregates in there from the MHTT. In the PCR, there's a significant decrease in the HTT expression as well. And in the Western blotting, of course, in the treated mice, that protein band is getting lighter compared to the untreated mice. So in HD knock-in pigs, abnormal gait and shorter stride length and just bad motor function is expected. However, in treated pigs, the stride length improved, the gait became more regular, they did well on the treadmill. As in our picture G here, it shows the footprint analysis on the sandy path I mentioned earlier. The untreated pigs, you can see the footprint is very irregular. The path is like all over the place. And you can see our poor friend is kind of stumbling over because it's expected that their coordination is not good. So in the treated pigs, you can see the path is like linear. The gait is so regular, it seems almost normal. In the graph on the top right, they just want to show the significant increase in the stride length in the treated pigs. And in the bottom, they just wanted to show that the body weight of these treated and untreated pigs did not change much. All it's trying to say is that it didn't really affect the overall health of these pigs while they're going through the treatment. Um, so on the pathology side, it did show healthier neurons compared to untreated pigs and reduced gliosis, which is consistent with the result we got in mice. So just to summarize, the purpose of this study was to see if CasRx can reduce mutant HTT in both mice and pigs in order to find any promising treatment for Huntington's disease. So their finding shows, improved motor function at different stages. They show protective neurons. They show a reduced harmful brain inflammation, partially restored gene expression. Um, so the result of the therapy were consistent across all ages and in both animal. So this shows that CasRx knockdown can improve the overall Huntington disease symptoms. I think most importantly, though, it works in large animal, which has never been done before. So a study that show like multiple animal model is really impressive. Because it's more conclusive compared to any other research we've found so far. And pigs has more similar brain physiology to human compared to mice. So that kind of mean like it's more feasible in humans. However, there is some limitation to this research that I've found. Um, so shown is some of the results compared to the pigs or animals that doesn't have Huntington disease at all. There was significant improvement in a lot of aspects, but it doesn't show a complete reversal as if like they were never sick in the first place. So that kind of suggests that maybe we need repeated treatment in order to get to that level. Um, for any long-term effects. Um, because unfortunately the mice and the pigs had to be euthanized for tissue analysis, which is very sad. Aside from that, it doesn't provide long-term data either. It doesn't show us any, you know, I'm sorry, disease progression or survival. It doesn't guarantee any permanent therapeutic effect. So I think what I would like to see going forward is maybe just longer term studies are needed to see if like repeated treatment in human could cause any adverse effect. Um, Overall, though, this research it shows pretty prominent result, and it was pretty consistent throughout all of the tests. It directly answered, questions of like the effectiveness of CRISPR-Cas9 system in larger animal. Um, you know, the results were consistent even in behavioral outcomes. I think not only that, but it preserved neuron function. It doesn't affect their health. And in my opinion, this opens up a lot of avenue for treatment and possibility of maybe um, future research on dosing, maybe long-term follow-up, and maybe even the possibility of combining CasRx with other therapeutic strategies so that we can improve our treatment in Huntington disease. And that concludes my presentation. Um, Here is my references, and I hope you guys enjoy. Thank you.